at that cellular level. Remember how I said in the very beginning, everything was going to happen at the cellular level? It does. So, at the cellular level, does that red blood cell that is traveling through that capillary, does it get to leave that vessel, the capillaries? No. What it is doing at this level, it's passing through this capillary bed at one cell a time. Now think about your blood flow. Up here, aorta, huge, massive amount, lots of the red blood cells. By the time it gets to a capillary, the capillaries are so small, red blood cells can only pass through one cell at a time. Not only do they pass through one cell at a time, they're getting squeezed. The squeezing allows oxygen to be pushed off. But the problem is, these cells that are behind, that we're feeding to, those cells can have those mitochondria that are using the materials, and the waste that's been given off is my CO2. Is everybody following me? So now CO2, which will be out here in my extracellular fluid, it needs to move into my capillaries so that it can go by way of the veins back to the heart, make its way to the lungs, and we breathe it out. Is everybody following me? But now, CO2. Let me think here for a second. How many of y'all drink sodas? They have carbonic acid in them. Why? Okay.
What gives it like that bubbly?
Do what, Nick?
Is that what gives it that bubbly thing? So
the fizzy bubbly.
Yeah.
If y'all know anything about movies. Okay, the fizzy bubbly. All right? Because what do we see at a drink machine? We could see it. Okay, it's behind the scenes. When I was growing up, you got to see it. It was so fun watching somebody hook up that CO2 tank to the drink machine. Oh yeah, that was hilarious. Yeah, anyway. Okay, so now we have everything behind the scenes. The CO2 is a gas. Ding, 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 ding. What is the CO2 in our cell? It's a gas. And that gas, just like a drink machine, okay, is being put into a fluid, the soda mix, giving it fizzy bubbly and carbonic acid. So now, if I'm at my cellular level and the CO2 that has been created because the cells had to make energy, that CO2 has to make its way into my bloodstream, i.e. at the level of the capillaries. That is now a gas, and what is our blood? Alex?
Liquid.
A liquid that hopefully has about a 75% water content, i.e. the soda mix. So now I have put that gas into the fluid, and I have made it fizzy bubbly, so to speak. But what I have done is it has now received carbonic acid. Acid. Acidic. What does it make you think about?
pH.
Okay, pH. We said, I think, I don't know if it was the other day or a couple days ago, we have to make sure the pH of the blood remains stable. Yes? Okay, so I know that I have a pH of blood that needs to remain stable. But I just added CO2 to the fluid that can make it acidic. My red blood cells can help with that. Is everybody kind of following me? Okay. So, yes, the red blood cell can pick up CO2, and it will. But the majority of the CO2 released by the cells that are at that tissue level of the capillary, it's going to need to be buffered so that it can actually be transported in the blood, not on the red blood cell. So the majority of our CO2 from the cells has to travel buffered in the bloodstream. So one of the things that my red blood cell is going to be able to do in this red blood cell, because it has spit out all of its organelles, the only way it gets energy is anaerobic, meaning that process of glycolysis. And in the cytoplasm, where we're going to find our hemoglobin, that protein, okay, we're going to also find an enzyme. That enzyme is called carbonic anhydrase, because what did I make? I made carbonic acid. And if it's an enzyme and it ends in ASE, it's got to do something to that carbonic acid. And it will. What it will do is it will act on that carbonic acid molecule and create what we call a bicarbonate ion. It is now buffered. It is not acidic. It will make its way, once at that capillary level, it'll make its way through the blood traveling through the vein, back to the heart, which we'll learn about, go to the lungs, and at the lungs, at that level, it'll get dropped off from that structure, and we have our CO2 in the lungs, and breathe out, and no more CO2. And by doing this, it allows for the blood, the pH, to remain stable. Is that not cool? Y'all ever thought about your red blood cells? Kind of cool. Just saying. Now, other parts about this little red blood cell, for something so tiny, okay? The plasma membrane. Ah, the what? What's the structure of a plasma membrane? They're embedded into it. They're embedded in the plasma membrane. Nate?
Does it have the phospholipid?
It is our phospholipids, known as that phospholipid bilayer. Does everybody remember that? So, that plasma membrane phosphate head, lipid tails, in a bilayer. And then, if you remember, because I've got the video up, okay, it has proteins that are embedded into it, and those proteins work to do something for the structure of our red blood cell. One of the things we find on this phospholipid bilayer, we find glycolipids. Do you remember how the structure of a plasma membrane would show stuff exiting off of it? Okay. The reason on the red blood cell that we have these glycolipids, it determines blood type. So those glycolipids that will be extending off of the plasma membrane help determine A, B, AB, O for the blood. Some of you, I think, know your blood type. Okay? I know, I need to go find out. But anyway, so that structure is what determines it. The other thing about the red blood cell, do you guys remember the cytoskeleton components of the cell? Okay, because we are animal cells, we're able to be squishy. If we had a rigid cell wall, we wouldn't be able to have the movement that we have. Okay, we'd all be very rigid. All right, but being an animal cell, we're squishy. Our cells literally get to and come back up. What did I say needed to happen to the red blood cell at that capillary level? It had to squeeze through. So the structure helps that red blood cell squeeze through the capillary. That is so cool. At least I think it is. So let's take a peek real quick back at the red blood cell production. Erythropoiesis. Do you guys remember hematopoiesis, where we talked about the stem cell as a whole? Okay. Well, now that I need to give rise to my red blood cell, the eryth, okay, that would be erythropoiesis. So, just like we said the other day, we have all together the one big stem cell. Now, to give rise to the red blood cell, do you guys remember those colony-forming units that would form? So we form a colony for erythrocytes, an erythrocyte colony-forming unit. From there, it will give rise to my blast. Do you guys remember? Blast, sites, clast. Okay? Blast is immature. So the first one it gives rise to is my immature red blood cell. Note, huge nucleus. Why? Why is the nucleus so huge?
The DNA is hot.
And based on that DNA, what is it focusing on making?
Proteins.
Specifically what?
The cytos, the plasma membrane.
Alex?
Hemoglobin.
The hemoglobin. And we got to make that little carbonic anhydrase one too. So that nucleus is huge because it's only going to keep it